Welcome to the Wonder Fungi Podcast. Today, we are playing doctor.
We certainly are. And I want to be super clear right from the start. We are not playing checkup.
Right.
This isn't a take 2 aspirin and call me in the morning kind of thing. We are talking about high-stakes, sterile trauma surgery.
It's such a bold way to frame it because, your first thought with mushrooms is farming.
Mm-hmm.
You think of like dirt and rain and nature. It feels messy. But looking at the sources we've got for today, and it is a heavy stack of them, the language is anything but agricultural. It's all medical.
It's completely medical. I mean, we're looking at papers on bacterial endospores that you'd find in a hospital sterilization protocol.
Seriously.
Yeah. And fluid dynamics papers on gas exchange and partial pressures. We have these precision protocols for inoculation.
Yeah.
So if you go into this thinking it's gardening, like you're planting tomatoes where you just throw seeds in the dirt and hose them down,
You're going to have a bad time.
you are going to fail. The sources are absolutely unanimous on this one point. If you treat mycelium like a plant, it dies. You have to treat it like a patient on an operating table.
And the whole reason for that super clinical approach, is the room we're sitting in right now.
Exactly.
I was reading one of the sources you sent. I think it was the Swiss Bay one, and it describes the air around us as the invisible war. It's genuinely unsettling.
It is an invisible war, and that's a concept that's really hard to, properly internalize. Right now, wherever you're listening to this, your living room, your car, doesn't matter, you are swimming in a soup.
Mm-hmm.
The air is an empty space.
No.
It is a suspension of particles.
The invisible cloud.
That's it. And we're not just talking about dust. The source material breaks it down. We're talking mold spores, bacteria, viral loads, just stuck to skin cells, pollen. And the critical thing here is the size.
Right.
We're talking mostly between like 0.5 and 20 microns. To give you some perspective, a red blood cell is about 5 to 10 microns.
So we're talking about like biological shrapnel, just totally invisible to the naked eye.
Completely invisible. But gravity's the great equalizer here. Anything bigger than about 10 microns, it eventually gets pulled down. And that means every single surface in your home, your counter, your clothes, your hair, the jar you're about to use, is covered in this fallout.
It's coated in potential infection.
Yes.
Okay, so let's set the stage properly for this deep dive then. You, the listener, you are the surgeon. The grain bag or your jar, that's your patient. And this invisible cloud is the infection, just waiting to get into the wound. And one slip up, one tiny little gap in your protocol, and the patient, your mycelium, it dies.
That's the tension. And that brings us really to the core mission of today's deep dives. We have to navigate this paradox.
Yeah.
We have to keep this aggressive, invisible infection out. But at the very same time, we have to keep our patient breathing.
The suffocation versus contamination balance.
It is the central conflict of all mycology. Seal the patient up too tight to keep the germs out, and you suffocate them.
But if you open them up to let them breathe?
You let the infection in.
So we're going to untack that paradox completely. We're going to look at the protocol of fire, which sounds super intense, but is actually just, good physics.
It really is.
We're going to dismantle the more is better myth, which I really think is the #1 killer of beginner grows.
Oh, easily.
And we are going to explain why a roll of, paper tape from the pharmacy aisle might just be the most advanced piece of engineering in your entire setup.
And I think we should probably start where every surgery starts.
The incision.
Or, in our case, the needle.
The sizzle.
The sizzle.
Okay.
Wait, let's walk through this scenario.
Yeah.
You've got your syringe. It's full of spores, or maybe liquid culture. You are ready to inject it into your grain bag. You're probably a little nervous.
Yeah.
And the precision protocol source is super, super strict about this next step. It calls it the red hot rule.
This is non-negotiable. You have to heat the tip of that needle until it is physically glowing red, not just hot.
Glowing.
Glowing red. So why is the color so important? I mean, isn't hot enough to kill a germ?
Well, hot is subjective, isn't it? You might think 100 degrees Celsius is hot, but as we're going to see later, some bacteria basically treat boiling water like a warm bath.
Wow.
Red is a temperature benchmark. When steel glows red, you're looking at temperatures over 700-800 degrees Celsius. At that temperature, organic matter doesn't just die, it disintegrates, it's incinerated.
Yeah.
We're trying to burn off any bacteria or mold spores that might have just landed on the needle from moving it through the air.
Okay, so total incineration of the enemy. But here's where the Swiss Bay source adds this layer of horror to the story. because there's a really specific mistake that happens right after the flame goes out.
The horror story. And it happens to almost everyone at least once.
Okay, picture this. You have heated the needle. It's glowing this angry red. You're anxious because you know the invisible cloud is all around you. You just want to get those spores inside the sterile bag as fast as possible. So you immediately plunge that red-hot needle into the injection port.
And you hear it. That hiss. That's the sound.
And I think most people hear that and they think, oh, nice, sterilization in action. I just fried anything bad.
And they would be so wrong. That hiss is the sound of surgical failure.
Okay, explain that. What is physically happening when you hear that hiss?
Two catastrophic things are happening. The first, and the most obvious one, is that you're cooking your own soldiers.
Right.
You're injecting biological material spores, mycelium, through a steel tube that's hundreds of degrees hot. You are boiling them alive before they even get into the bag.
So you're just injecting a ghost. You've killed the patient before the surgery even started?
Pretty much, yeah. But the second thing is way more subtle. And according to the fluid dynamics papers we looked at, maybe even more dangerous when it comes to contamination.
Okay.
The Swiss-based source calls it an explosive burst of vapor.
An explosion.
A tiny one, yeah.
Okay.
A microscopic one. When that red-hot metal touches the moisture in the grain or the rubber of the port, it flash boils that water instantly. And that creates a sudden, violent expansion of steam.
And explosions create wind, they create shock waves.
Exactly. That burst of vapor makes a localized high-pressure shock wave. So if there are any mold spores just sitting near that injection site, maybe they survived the alcohol wipe, or maybe they just landed there a split second ago.
Oh, no.
That vapor burst liberates them. It blows them into the air. It creates this turbulent little aerosol cloud right at the point of entry.
So by being too impatient with the heat, you're actually helping the enemy. You're blowing them right into the wound you just made.
You're literally creating a vector for them.
Yeah.
You're using their favorite way to travel.
So what's the remedy? The protocol of fire can't just be get it hot.
No, the protocol is get it hot, then wait. The sources all say you need a cooling period of about 3 to 5 seconds. You just need that metal to get back to a temperature that won't flash boil water on contact.
I've also seen people mention squirting a little bit of the liquid out first.
Yeah, that's a totally valid technique. The precision protocols mention it. If you push a tiny, tiny amount of the solution out of the needle before you pierce the bag, that liquid cools the metal down from the inside.
But then you lose some of your spores.
You sacrifice a single drop to save the entire culture. It's just the cost of doing business.
Yeah.
It makes sure that when you enter the patient, the tool is sterile but not destructive.
Okay, so we've navigated the red hot rule. We waited, no hiss. The needle is inside the bag. Now we get to the next trap. And I think this one is purely psychological. It's the more is better fallacy.
Oh, this is classic human psychology. It's like the American supersize mentality applied to microbiology.
But it makes sense on the surface. If I'm a beginner, I'm thinking, I want mushrooms fast. Spores make mushrooms. So 5 cc's of spores will make mushrooms 5 times faster than 1 cc. It feels like linear logic.
It is a logical thought process, but it's biologically a disaster. The biology here isn't linear at all. It's all based on thresholds.
Why? What's happening when we inject too much?
Because you're not just injecting spores, you are injecting water.
Right.
The spores are suspended in a liquid. And this brings us to what I call the physics of the swamp.
The swamp.
So in our sources, especially the bacterial contamination paper, they talk about moisture content with, like, extreme precision. Mushroom mycelium is Goldilocks. It doesn't want it dry, but it absolutely positively does not want it wet.
So what's the number? What's the limit?
The danger zone is anything over 60 to 65% moisture content within the grain.
That seems incredibly specific. How would a home grower even measure that?
Well, you usually rely on the squeeze test when you're preparing your grain. If you squeeze it and water drips out, it's too wet. But here's the trap. The Precision Protocols source points out that beginners often inject 2 to 5 cc's of liquid per bag.
Right, because you buy a 10 cc syringe, you've got 2 bags, you just do the math, 5 cc's each.
Exactly. But if you're using, say, a standard ready-to-eat rice bag or a jar of grain that's been prepared correctly, that grain is already hydrated. It is already sitting right at that optimal 60% limit.
So when you dump another 5 cc's of water in there...
You are drowning the patient. You're pushing the moisture content way past the tipping point, probably up to 70–75%. And then gravity takes over and all that extra water pools at the bottom of the bag.
Ah.
And that pool of water is not a swimming pool for your mycelium. It's a breeding ground for the villain of our story.
Bacillus.
Bacillus. also known as wet spot or sour rot.
I love that the sources get into the sensory details of this contaminant, because if you've ever smelled it, you never, ever forget it.
No, it's so distinctive. It activates a really primal disgust response in you.
The source lists the smells as dirty socks, vomit, or sour fermentation.
And visually, it's just as bad. You get these dull gray or like brown patches pressing up against the glass or the plastic. The Gromagic source describes a slimy texture and a melting appearance.
Melting mushrooms is a truly horrifying image.
It is. The bacillus produces this slime matrix, a biofilm, that just coats the grain. And here's the critical point. Mycelium can't colonize slimy grain.
It can't.
It's a physical problem. It can't grip it. It can't digest it. The slime is like a force field.
So that's where you get these uncolonized blind spots that the zombie mushrooms article was talking about.
Exactly that. You look at your jar, and most of it's beautiful and white, but there's this one patch at the bottom that the mycelium just will not touch. It treats it like a quarantine zone. That's because invisible bacteria have already claimed that territory. They've put up a slime wall.
And this all started because we thought more is better and injected too much fluid. So what's the right amount? If 5 cc's is a flood, what's like a spring shower?
The precision protocols are very, very clear on this.
Yeah.
0.5 cc's to 0.75 cc's.
That's it.
That's it. It's less than the milliliter. We're talking a few drops.
That just doesn't feel like enough to get millions of spores in there.
But it is. In a proper spore solution, there are millions of spores per milliliter. And you only need 2 compatible spores to meet and germinate to start the entire process. You're introducing genetics, not providing irrigation. The grain already has the water it needs.
It's amazing how little you actually need. It really reinforces that whole surgical idea. Precision over power.
That's it.
You're not fire hosing the patient, you're just administering a very precise dose.
Precision is everything. You have to trust the biology. The mycelium knows how to grow. You just need to give it the spark without, putting out the fire with a bucket of water.
Okay, so we've avoided the hiss. We've used the right amount of liquid, just half a cc. Now we have to talk about the environment inside the bag. This brings us to the suffocation trap.
And this is where we have to get a little deeper into the biology. We need to understand the fundamental difference between our hero and our villain.
The hero being the mycelium.
Yep. Specifically, Psilocybe cubensis in the context of the comprehensive analysis source we've got. And the villain is our old friend Bacillus.
So what's the big difference? I mean, they both eat grain. They both like warm, dark spots.
The difference is respiration, how they breathe. And this is where the whole doctor metaphor really holds up.
The source calls the mycelium an obligate aerobe. That sounds serious.
It is. Obligate is the key word there. It means it's not a choice. It must have oxygen. The source explains that the mycelium needs oxygen to create ATP adenosine triphosphate.
That's the fuel for the cells, right?
The very fuel for all life, pretty much. Without oxygen, the electron transport chain inside the fungal cells literally just halts. It shuts down.
Yeah.
The source calls this metabolic stalling.
So it's basically holding its breath.
It is. And while it's holding its breath, it stops growing. It stops producing its own antibiotic compounds. It stops fighting. It just sits there, completely vulnerable.
And meanwhile, the villain.
Bacillus is a facultative anaerobe.
That sounds like a superpower.
In this context, it basically is. Facultative means it's adaptable. It can survive with oxygen, sure, but it can also switch modes. If the oxygen levels drop, it doesn't stall. It switches to fermentation.
So while the mycelium is choking, the bacteria is just switching gears and carrying on.
It's thriving. In fact, a lot of these bacteria actually prefer low oxygen environments because it gets rid of their competition, the aerobic organisms like your mushroom mycelium.
Okay, this brings up a really important point about the sealed bag disaster, because I think intuitively, if you're trying to keep contaminants out, like we said, the air is full of enemies, your first instinct is to seal the bag up tight, make it totally airtight.
It's the Tupperware logic. I want to keep my food fresh, so I seal it.
Yeah.
But in this surgery, that logic is fatal.
Because if you seal it airtight,
You're going to deplete the oxygen. The comprehensive analysis source breaks this down beautifully. As the mycelium starts to grow, even just a little bit, it consumes oxygen and it releases CO2.
just like us, we breathe in oxygen, we breathe out CO2.
Exactly like us.
Yeah.
So inside a totally sealed bag, the oxygen levels are just plummeting and the CO2 levels are skyrocketing.
The source mentioned that if the CO2 gets too high, something like above 20,000 parts per million, it creates carbonic acid.
It does, yeah.
Yeah.
It starts to acidify the substrate. But the really immediate problem is just the lack of oxygen. The mycelium stalls. It stops defending itself. And now you have created a warm, wet, low-oxygen environment.
You've built a paradise for bacteria.
You have literally rolled out the red carpet for bacillus. They switch to fermentation. They start multiplying like crazy. They produce that sour smell we talked about, which is literally the byproducts of fermentation. And suddenly your bag is rotting instead of colonizing.
So the aha moment here is that by trying to suffocate the mold, you are actually suffocating your patient and actively helping the infection take over.
Precisely. You cannot starve the bad guys of air without killing the good guys first.
So we need a hole.
We need a hole.
But a hole is an open door for that invisible cloud of spores we started with.
Yep.
And that right there is the paradox, the micropore paradox.
And this brings us to the surgical solution. And I just love that the hero product here is something you literally buy in the pharmacy aisle.
3M micropore tape.
Medical grade paper tape.
It is the unsung hero of amateur mycology. It truly is.
So how does a piece of paper tape solve a paradox? I mean, when you look at it, it just looks like tape. It looks flimsy.
It's so deceptive. If you look at the micropore filter tape source and the Booming Acres breakdown, they explain the physics of it. It's not just a screen. It's not like a window screen where small things can fly through if they're just small enough.
It's depth filtration.
Exactly.
Yeah.
Think of it like a labyrinth. It's a 3D maze of tangled fibers layered on top of each other.
Okay, I like that image. A maze.
So when air passes through, the gas molecules, the oxygen, the CO2, they're tiny. They can navigate that maze really easily, just bounce off the walls and flow right through. But a mold spore, or a dust particle that's carrying bacteria.
They're like giants in this maze.
To them, that tape is a dense forest of tangled roots. They physically crash into the fibers. They get stuck. They get trapped by the electrostatic charge of the material. The source mentions a filtering efficiency of up to 99% for contaminants, even down to that 0.5 micron level we were worried about.
So it lets the breath out, but it keeps the germs out.
It creates a selective barrier. It solves the paradox.
But there's another layer of physics here that I find just absolutely fascinating. It's not just about the tape. It's about what's happening inside the container that's pushing the air through the tape in the first place.
The chimney effect.
Yes, this is pure thermodynamics.
It's beautiful engineering by nature itself.
I remember reading this in the comprehensive analysis. It says the mushroom actually generates its own heat.
It does. It's a living metabolic process. Breaking down grain is work, and work generates thermal energy. So inside your jar or your bag, the temperature is actually a few degrees warmer than the air in the room.
And basic physics. Warm air rises.
Warm air rises. So this warm CO2-rich air that's being generated by the mycelium naturally rises up to the top of the bag.
Where the filter is.
Exactly, where the filter is. It pushes out through the micropore tape. And because nature abhors a vacuum, as that warm air leaves, it creates a slight negative pressure that pulls fresh, cool, oxygen-rich air in through the tape.
It's breathing.
It is breathing. It's a passive lung. You don't need fans. You don't need pumps. You don't need any electricity. You just need to respect the thermodynamics and give it the right filter.
That is unbelievably cool. It makes the whole organism feel so much more alive. It's not just a fungus sitting there. It's an engine.
It is an engine.
Yeah.
And it's engineering its own environment if you just let it.
Now, we have to talk about the competition here. The old ways. Polyfill.
Ah, yes. Polyester pillow stuffing.
The Booming Acres source goes hard on polyfill.
It does, and for very good reason. Before micropore tape became the standard, people would drill holes in their jar lids and just stuff them with polyester fiber from a pillow or a craft store.
Which works. I mean, theoretically, it's a filter.
Theoretically. But the source points out all the fatal flaws. It's inconsistent. How tight do you stuff it?
That's the problem. If you stuff it too tight.
No air. Suffocation.
And if you stuff it too loose.
Contamination. The bugs just fly right through. It's a total guessing game.
And it's messy.
It's such a mess. Little strands get everywhere. Moisture can wick right through it.
Yeah.
You can't really clean it. Tape is binary. It's on or it's off. It's engineered to a specific standard. 3M spent millions of dollars developing that pore structure in a lab. You stuffing a chunk of a pillow into a hole is, well, it's less engineered.
It's set it and forget it versus hope for the best.
Exactly. And in surgery, we do not like hope. We like certainty. We like standardized protocols.
Okay, so let's take all this theory, the flame, the liquid volume, the tape, the thermodynamics, and apply it to a real-world scenario, the Capri Sun technique.
This is a fantastic example of practical ingenuity. It's described in the Precision Protocol source, specifically for those ready-to-eat rice bags. But the principles, They apply to everything.
And it's called Capri Sun because, well, it's exactly like sticking a straw in a juice pouch.
It is. So let's walk through the surgery. Step 1, you've got your bag. You've got your flame-sterilized, cooled-down needle. You inject right through the center of the bag.
Why the center, not the top or the bottom?
Distribution. You want the colonization to start in the middle and work its way out radially. It's just the most efficient path.
Okay. You inject your 0.5 cc, not 5 cc's.
A crucial distinction, just a sip.
Now here's the twist. Step 2. The source says, clip the corner.
This is the critical step that most people are so afraid to do. You take a pair of sterilized scissors and you physically cut a corner off the bag.
You're cutting open the patient. That just feels wrong. We just spent 20 minutes talking about keeping germs out of an open wound.
It feels totally counterintuitive. You're creating this massive wound, but it's a dedicated gas exchange port, a GE port.
Because the little needle hole isn't enough.
That needle hole is tiny. It seals up behind itself almost instantly. And you have to remember, the mycelium is an obligate aerobe. As it grows, its demand for oxygen increases exponentially. That little pinprick is not going to provide enough airflow for the chimney effect to really work. You'll bottleneck the gas exchange.
So you clip the corner to make a bigger vent, but now you have this gaping hole.
Which brings us to step 3. Seal it immediately with micropore tape.
And this is the balance we were talking about at the start.
Yes. You've created a large opening for air, solving the suffocation problem. But you've covered it with a medical-grade depth filter, solving the contamination problem.
It's a closed system, but it's breathable.
It's elegant. It uses the physics of the tape and the biology of the organism to create the perfect little incubator.
So you've done the surgery, you've been precise, you've taped it up. Now comes the waiting game, post-op care.
And this is where observation becomes your key diagnostic tool. You can't interfere anymore. You can only watch.
So we need to know what to look for if the surgery failed.
Right. Because even with the best protocols, sometimes that invisible cloud wins.
The zombie mushroom source has a whole checklist of symptoms. We already mentioned the smell, the dirty socks, and the slime. But what are the visual cues before the slime really takes over?
Well, one of the earliest signs you can see is in liquid cultures, or even in the moisture against the glass of a jar. If it looks cloudy or milky, that's a very bad sign.
A bacterial bloom.
Yes. Healthy mycelium water should be basically clear. If it looks like skim milk or if it's turbid, that's bacteria just exploding in population.
And we talk about the blind spots.
That is a huge tell. If you see the white mycelium racing through the jar, but it completely avoids one specific area.
Like there's a force field there.
It effectively is a force field. It's avoiding a bacterial stronghold. The bacteria are there, you just can't see them yet, but the mycelium can sense them chemically, and it is staying away. It's retreating.
Now, I want to circle back to something we touched on in the intro, because it's genuinely the stuff of nightmares. The endospore threat.
Ah, yes. Bacillus endospores, the ultimate survivalists.
We talked about how flame sterilization kills things on the needle, but the expert in the bacterial contamination source mentioned that these endospores are incredibly resilient inside the grain itself before you even start.
Resilient is such an understatement. They're time capsules. When bacillus bacteria feel threatened, say they run out of food or moisture in nature, they form these hard armored shells around their DNA. Those are endospores.
And they are heat resistant.
Extremely. They can survive standard boiling water 100 degrees Celsius. They just laugh at it. They can sit in boiling water for hours and just stay dormant.
That is terrifying. So if I just boil my grain on the stove...
You might kill all the active bacteria, sure, but the endospores will survive. Then when the grain cools down and you introduce that little bit of moisture from your syringe, they wake up, they germinate, and suddenly you have a massive bacterial infection from a supposedly sterilized jar.
So how do you kill them? If boiling doesn't work, what does?
You need physics on your side again. You need pressure. The source is very specific. 121 degrees Celsius at 15 psi.
Pounds per square inch. That's autoclave standard.
And pressure cooker standard. That extra temperature, which you can only achieve under pressure, is the only thing that cracks that armor. It denatures the proteins inside the spore.
It really emphasizes why that pre-op preparation is so critical. You just can't wing it.
No, sterility is not a gradient. It's absolute. You are either sterile or you are contaminated. There's no such thing as mostly sterile.
That's a great mantra for this whole deep dive. There is no mostly sterile.
It's the surgeon's creed.
So let's just recap what we've learned today. We've gone from thinking about this as farming to treating it like surgery.
We've established that the air itself is the enemy. an invisible cloud of contaminants just waiting to fall.
We learned the protocol of fire. Get that needle red hot, but wait for the cool down to avoid the hiss and that vapor burst that actually spreads the enemy around.
We busted the more is better myth. Half a cc is plenty. Don't create a swamp. Don't drown the patient in their own bed.
We identified the villain, bacillus or wet rot, and our hero, the mycelium, the obligate aerobe.
And we've really understood that the whole battle is between suffocation and contamination. If you seal it, they suffocate. If you open it, they get infected.
And the solution is the micropore paradox, a piece of tape that's a maze for germs but a highway for air.
And it's powered by the chimney effect, the mushroom's own body heat driving its own ventilation system.
It's a beautiful system when you actually understand the mechanics of it.
It is. It transforms the hobby from just a guessing game into a science. You stop asking, did it work? And you start understanding why it works.
Or why it didn't.
Or why it didn't, which honestly is often more valuable.
Before we go, I want to leave our listeners with one final thought. We've talked a lot about the fragility of the mycelium, how easy it is to kill it, how easy it is to contaminate it.
It does seem delicate.
It does. But then you think about that chimney effect again. This organism, even when it's just a tiny patch of white fuzz, is generating heat. It is changing the air pressure around itself. It is actively pulling in the fresh air that it needs to survive.
It's not passive at all.
It's not.
Yeah.
It is fighting for its life. It's working. It is alive and breathing before you can even see it.
And if you just give it the right filter, if you just give it a fighting chance against that invisible cloud, it will do the rest. It wants to grow.
It wants to grow. You just have to be the surgeon that clears the way.
Precisely.
Thanks for listening. Happy growing. And don't forget to subscribe.